Hi everyone, welcome to this M2D2 talk today. Um, we'll be exploring the topic of equivariant scalar field for molecular docking with fast Fourier transforms. And our speaker today is Bowen Jing. He's a third year PhD student in the Electrical Engineering and Computer Science department at MIT. And he is co-advised by Tommy Yakola and Bonnie Berger and works on deep learning for structural biology and drug discovery, with a focus on generative models and molecular modeling. Thank you so much, Jean, for kind of coming here today to present your work. And yeah, looking forward to it. Let's jump into it.
All right, good morning, and thanks for the opportunity to speak at this reading group. Let me share my screen. And let me move the video bar down here. Okay, is it all working?
Yes.
Okay, all right. So good morning, everyone. Today I'll be talking about our work on developing a machine-learned scoring function for molecular poses in a way that allows for very fast optimization over rigid body degrees of freedom using fast Fourier transforms. So the task that we are focusing on, just to set the stage for what we're going to talk about, is that of blind molecular docking. So given a small molecule ligand specified as a molecular graph and the 3D structure of a protein target, we're going to want to find the structure of their interacting complex. So this is just a more formal way of saying, specifying the inputs and the outputs of what we want to do. And specifically, our outputs in the general blind docking task are both the conformation of the ligand, because our inputs does not contain information about the 3D conformation of the ligand, but rather only its chemical conductivity, and also its exact position and orientation. So its specification over the rigid body degrees of freedom. Now, the way that this is typically done before the deep learning era, as many are likely familiar with, is by viewing this problem as an optimization problem over the space of molecular poses. So in this optimization problem, you typically have a scoring function, which tells you how good a pose is. And this is typically decomposed as the sum of pairwise energy terms, where the pairs are all the possible protein ligand interaction pairs. For each of these, you have either a penalty or a positive score, and then you sum over all of these pairs to obtain the scoring function for the pose. You then have a whole host of possible optimization algorithms for optimizing this scoring function, the details of which I will certainly not have time to cover today, but there's been decades of literature on combining various options of scoring function and optimization algorithms to try to find the best possible pose in the fastest manner possible. And as you can imagine, this process can be quite slow. Now, what we would like to do is to take an alternative approach for how to parameterize this scoring function, and in particular, how to learn a scoring function that might be optimized in a faster manner. And the way this parameterization is going to be developed is going to be inspired by what is already actually standard practice in protein-protein docking. So let's take a quick detour and look at how protein-protein docking is formulated. So at a high level, the protein-protein docking problem seems very similar to the protein-ligand docking problem. You're trying to optimize over certain degrees of freedom, and typically there's some implicit energy function that you're trying to optimize. But in protein-protein docking, an approach that has become quite popular and is used as a subroutine in many well-known software packages is something like the following. So you parameterize your scoring function not based on a pairwise sum of energy terms, but rather as a overlap integral between functions defined over the volume of the protein in three-dimensional space. So here's an example. So let's say that you have these two proteins that you want to dock, and you define a function a of x, which is +1 on the surface of this first protein, and b of x, which is +1 on the surface of the second protein. And by the surface here, I mean maybe within like a 3 or 5 angstrom radius of the surface. You can then say that the shape complementarity of a particular pose is given by the overlap of, or rather the integral of the product of these two functions, right? So in the diagram shown here, the two proteins are not interacting at all, and so the integral is 0. But if they dock against each other in the manner suggested by the figure, then we would have some region where a times b is +1, and you would get a positive integral. You can also have another term that penalizes the steric clash of the two proteins when they interact. So to do that, you define a new function c of x, which is +1 in the interior of the first protein, and another function d of x, which is -1 in the interior of the second protein. And this is actually a typo here. So in the second integral, if you integrate the product of if you integrate the product of b times d, where d is offset by some translation t, then that will tell you the amount of steric clash between the two proteins when the second protein is translated by t. So what you can then do is you can say my overall scoring function is simply the sum of these two functional inner products, right? One which expresses the amount of shape complementarity, and the other which expresses the amount of steric clash. And the sum of those, I can say, should be very high for a good pose and low for a bad pose. The nice thing about this parameterization of scoring function is that with the fast Fourier transform, it is actually possible to evaluate the value of this scoring function for all possible translations of the two proteins relative to one another. And this simply relies on the fact that the scoring function, the way that we have written it, is just the sum of convolutions of pairs of functions. So convolution of a and b plus the convolution of c and d gives us the desired scoring function as a function of the translation of the second protein, right? So if we evaluate this Fourier transform, we get all of these integrals for free, essentially, and we can go directly to the argmax of that function once we have evaluated this Fourier transform over a multidimensional array in our memory. So this is actually a very common subroutine in protein-protein docking. And what we would like to explore is how can we use this for protein-ligand docking? Now, the general case in protein-ligand docking, why have people not used this technique? Well, it's partially because for protein-protein docking, most of the interactions or most of the scoring functions that people like to use are based on shape complementarity anyways. But in protein-ligand docking, the situation is a bit more complicated. The pairwise scoring functions that I showed on the previous slide generally have functional forms that are a little bit more complicated. Actually, do I say this here? Yes, okay, that's what this slide was about to say. So, in the protein ligand case, you can also imagine that we should be able to write down the scoring function as a convolution of this type, or rather, in this case, it's not a convolution, but just this integral of the product of the two functions. But the problem is that standard protein ligand scoring functions that are based on these pairwise energy terms, they generally cannot be written down, or at least they cannot be easily written down as cross correlations of this form, which makes it hard to use these fast Fourier transform ideas to optimize the scoring functions that people actually use in practice. What we explore in this work is, if our handcrafted scoring functions are not well suited for this optimization procedure, why don't we try to actually learn a scoring function that is amenable to these fast Fourier transform-based procedures? And the way that we're going to do that, of course, is with machine learning, because if we don't know how to write down a function with the property that we want, we just do a billion steps of gradient descent to try to get such a function, right? So that's what we're doing in this work. We're going to use equivariant neural networks and try to parameterize a scoring function which has more of a cross-correlation type functional form rather than a pairwise pairwise energy sum functional form. Okay, so how do we do that? How do we parameterize this function? So what we're going to do is we're going to employ E3NN equivariant neural networks that are going to look at the input pose, right? So recall that the problem setting here is we have two rigid bodies and we're trying to find their best possible orientation relative to one another. And so the neural network is going to look at each one of those rigid bodies in isolation, and it's going to, in some fashion, output the scalar field that describes the interaction potentials with the other binding partner, right? So there's going to be one copy of the neural network that looks at the protein and one copy of the neural network that looks at the ligand. And in both cases, they're looking at the 3D structure of the protein and the ligand. So the way that this parameterization is going to work is as follows. So the value of the scalar field at a point x relative to the ligand conformer, which in this case is specified by the graph G and the ligand atom position X. So we're going to evaluate the value of the scalar field at an arbitrary point X. This is going to be decomposed into the sum of contributions from each atom. So N here is the atom index. For each atom, we're going to, around each atom, we're going to define a set of basis functions, which are going to be the product of the spherical harmonics given here, right? So you can see that this is the spherical harmonics evaluated where the origin is centered on each of the individual atoms in. And we're also going to multiply that with the set of radial basis functions, again describing the distance from that central atom xn. So for each atom n, we're going to have such a set of basis functions, which are indexed by j, l, and m, because the spherical harmonics carry indices l and m, and then we're going to choose j radial basis functions. And then each of those is going to have a coefficient, which is going to be the output of our neural network, right? So our neural network is going to do some message passing over the graph, and for each atom n, for each node n, it is going to predict the coefficients that should be attached to each one of these product basis functions, j, l, and m. So you can think of this as building up the scalar field atom by atom, where each atom you have some function parameterized in spherical coordinates around that atom, which is given by the spherical harmonics, as well as the associated coefficients with those basis functions. And then we're just going to sum over all of the atoms, right? So when you sum over all of the atoms, you will you will get a scalar field that looks something like this, where each contribution kind of mends or melds in with the contributions from the other atoms, and you just have this nice global scalar field for the molecule. The key point here is that this A function, this A neural network, the neural network which is responsible for outputting these coefficients, it needs to be SE3 equivariant, because when you rotate the molecule, you want your scalar field to transform in a predictable way. In fact, it should transform under a rigid body motion, right? And the only way in which that is going to be accomplished is if these coefficients A, C, and J, L, M transform appropriately under global 3D rotations. And this is something that is assured by E3 equivariant neural networks. So once we have these scalar fields, what can we do with them? Well, we can now evaluate the scoring function of all possible translations of a particular reference pose. So here, let's say that we have a reference pose where the protein atoms are at xp and the ligand atoms are at xl. And I want to explore what happens to the scoring function when I translate the ligand xl by some translation vector t. Well, this is going to just be the cross correlation of the two scalar fields, the ligand scalar field cross correlated with the protein scalar field. And to evaluate this cross correlation, we just need to evaluate the inverse Fourier transform. Well, again, this is just the convolution theorem at play here. We just need to evaluate the Fourier transform of the ligand scalar field and then the protein scalar field, multiply those two, and take the inverse Fourier transform. Each of which of these is in log n in runtime, which is the whole reason why this procedure is nice. Now, there's a nice trick for how to actually obtain the Fourier transform of the ligand field and the protein field. You don't actually need to, for example, evaluate the value of the field at each point over a grid and then apply a fast Fourier transform. You can actually obtain the value of the Fourier transform function directly. What we call the translational Fourier coefficients here. And the way that you can do this is by noting that the way that we have described our decomposition allows us to, well, I mean, I don't want to necessarily go into details for how this is derived. This is all in the paper. But there is a nice expression which just simply amounts to taking particular linear combinations of these AC and JLM coefficients, which are the outputs of the neural network prediction. So there is no point in this procedure where you actually need to evaluate the value of the scalar field at every point in space and then take the Fourier transform, because that would be quite expensive. So just to break down a little bit more what is going on in this expression, what we again see is a sum of contributions from each ligand atom n, or not necessarily ligand atom, but just each atom n. So the complex exponential here, as you might recognize, serves the role of translating in real space the contribution of that ligand atom n. And so the remainder of the expression is just the contribution for each ligand atom if it were centered at the origin. And as you might expect, the spherical harmonics show up here, and then the radial basis functions also show up here, but in a slightly different way. What happens to the radial basis functions is that they get applied under the spherical Bessel transform, which essentially is an important tool for doing spherical, or rather for doing Fourier transforms for functions defined in spherical coordinates, which is what we have here. And the spherical Bessel transform is essentially a decomposition of an arbitrary function over positive numbers into basis functions that look like this. So as you can see, it has a very Fourier-like flavor to it. And so it's not surprising that something like this might show up when you're trying to calculate the global Fourier coefficients for the scalar field that we've defined. Okay, so yes, was there a question? No? Okay. So now that we have the Fourier transform of the two scalar fields, one for the ligand, one for the protein, we can multiply them, take the inverse Fourier transform, and then we immediately get this cross correlation. Okay. So this is nice because we can optimize for a given reference pose the translation of that reference pose, right? But of course, there's more than translations. There's also rotations. So can we optimize over rotations immediately given a particular reference pose? It turns out that with this parameterization, we also can. And this is maybe a little bit less well known than the translational Fourier transform, but it's a body of literature that has been leveraged in many disparate fields, including crystallographic alignment of electron densities, as well as cross correlations of signals on the sphere, which is important in astrophysics, for example. So none of the math here is new, but I'm just going to present it again because I think the application here is pretty interesting and maybe a little less familiar than the translational case. So in the rotational cross correlation, what we desire is we have, again, a reference pose for the protein and for the ligand, and we want to ask what is the value of all the possible, or rather, what is the value of the scoring function for all possible rotations of the ligand from this reference pose? And what we're going to, so in the second expression here, I've just directly written that out. But we're going to also write that as a cross correlation. So this is not the traditional notion of cross correlation. This is just redefining the notation of cross correlation for this slide, just so that it's a little nicer to look at when we reference the function that we're trying to compute. So the way that this function can be computed, this rotational cross correlation, is as follows. So first we need to do a change of basis into a global spherical harmonic expansion of the scalar field. So what I mean here is that the way that we've defined our scalar field so far, as you can see on the previous slide here, is as a sum of contributions from each ligand atom. And from each ligand atom, we have a basis set which is in spherical coordinates centered at the position of that particular ligand atom. To do the translational, or rather to do the rotational cross correlation, we need to change basis into a set of spherical basis functions centered at a common origin. So, a common choice of origin might be the center of mass of the ligand, and that's what we typically employ in these experiments. So we're going to take the existing ligand scalar field, and we're going to just rewrite them in this global set of bases. And the computational procedure for doing this just essentially amounts to, well, it's just a change of basis, right? So you pre-compute some change of basis matrix, and then you apply that change of basis matrix to the previous basis coefficients, which were the A, C, and J LMs, and then you get the new coefficients, B, C, and J LMs. So this B network here is not the output of a neural network. This is just the you take the outputs of the neural network and then you apply a change of basis matrix to them. Okay, but then when you get these coefficients, what can you do? What you can do is you can immediately write down the rotational cross correlation of these two scalar fields defined by these two different BC and JLMs, right? One for the ligand, one for the protein, as a three-dimensional Fourier transform of some other function. And so, let's take a look at what that function is. So these DLMHs are these are essentially constants. Well, they're not constants, but these are things that you can pre-compute and are not related to the protein or the ligand. This is just the entries from the Wigner D matrix for rotations around the y-axis. So these are things that you can pre-compute, and you can just put them in your three-dimensional grid, right? And then the ILMs here contain the information about the two scalar fields. So you take the two scalar fields, one for the protein, one for the ligand. You evaluate this expression, and you get these ILMNs, which is a three-dimensional array of numbers, right? You get this three-dimensional array of numbers, and you multiply them with the three-dimensional array that you have from the Wigner D matrices, and then you apply a fast Fourier transform. You apply fast Fourier transform, and I should also say that the Greek letters here are indices for the rotations that you want to evaluate this cross correlation at, right? So this new function, this cross correlation will be a function of the Greek letters, right? After you sum over the Latin letters. So this is just a Fourier transform, and you immediately get another three-dimensional array which contains the value of the ligand scoring function as a function of the rotation of the ligand, right, relative to the reference pose. And you can think of those as the Euler angles of the rotation. But you immediately have this nice three-dimensional grid where all the possible rotations have been evaluated, and you can go to the you can look for the argmax of that array, and you can immediately optimize the ligand pose. Oh yeah, okay, I already said that. So the procedure that I've lined out so far is just once you have obtained the neural network that gives you those coefficients, how do you optimize the ligand pose over translations, and how do you optimize it over rotations? But then we have to ask, how do we actually obtain this neural network in the first place, right? So this is what we have. This is what I just described. And now we need a training procedure. So ideally, what we would like to do, and maybe what we can do, is we can train this as an energy-based model, right? Because normally training an energy-based model is hard because we don't have access to the partition function, because that's expensive to evaluate. But if you have the value of the scoring function or the energy for all possible rotor translations of the ligand, you can directly evaluate the partition function, right? It's just a six-dimensional array of numbers which you now have to sum. So that would be very nice. Unfortunately, we don't quite have it that nice because we cannot actually evaluate the scoring function for all possible combinations of rotations and translations. We can evaluate it for all translations separately, and then for all rotations separately, but we cannot combine the two. And so what we do instead is we view this as an energy-based model, first over the space of translations conditioned on a given rotation, and then an energy-based model over rotations conditioned on the space of translations. You can sort of think of this as a pseudo likelihood, right? Like the likelihood of a ligand pose should be the joint likelihood of its rotation and translation, but we cannot directly evaluate that. So instead, we evaluate the likelihood of the translation given the rotation, and then the rotation given the translation, and we try to optimize the sum of those two things. So that's going to be our training procedure. Once we've trained this network, we can look at the resulting ligand poses that we learn, or rather not ligand poses, but the scalar fields that we learn. So we train 5-channel scalar fields, and I'm just showing a couple of them here. So the first row is going to be the scalar field predicted by the neural network for the ligand conformer. And then the second row is going to be for the protein pocket. And in these particular illustrations, the pocket and the ligand have been aligned in their ground truth pose. So you can sort of look and see, okay, we can see some interacting features where the red and the blue, or the red features try to overlap with the red features, and the blue features try to overlap with the blue features. And you can sort of see that some of these channels are learning very simple features, like the one in the middle here, where the entire ligand is blue and the entire protein is red. This is like a steric clash channel, right? This is just trying to make sure that the ligand has no interaction with the protein, in terms of steric overlap. But then the other channels are a bit more complicated, and I'm not a chemist, so I don't want to be too speculative about what these scalar fields are representing, but they're certainly interesting to look at. Here again are some more. As you can see, again, the scalar fields, they are different enough from each other that there is reason to believe that each channel is learning separate information. But I will say that I observed improvements from going from 3 to 5 scalar channels, but not so much from 5 to 8. So it does seem to have maxed it out in terms of information carrying content at 5 channels. Okay, so now that we have the scalar fields, what do we do with them? So the original problem setup that I was talking about is, where you have the ligand specified as a molecular graph, and you want to simultaneously find its conformation as well as its rigid body positioning. So as you have, maybe have been able to gather from the talk so far, we're not actually directly addressing the first part of that problem statement, which is to find the conformation of the ligand pose. So that's a limitation of the method as it currently stands. And all of the ensuing experiments are going to assume that we want to dock rigid conformers, right? So you assume that you have a library of conformers for a given ligand, for example, and for each ligand conformer, you now want to find the optimal rototranslational placement of that conformer. What we can also do is a separate procedure where we have candidate ligand poses and just want to score them. In the scoring procedure, there's actually no fast Fourier transforms involved because the FFT is useful for optimizing the rototranslation, right, rather than directly scoring them. So I think I will just gloss over the details for the post scoring. They'll be on the slide, but I just won't talk about them in detail. Once we have these scalar fields, we can do docking in 2 ways, which roughly lines up with the 2 procedures that I've described so far for doing the FFT. We can either do translational FFT and rotational FFT. But when we do translational FFT, we need to search over the rotations by hand, right? So there's unfortunately still always a grid search component involved. We can either search over the rotations by hand and then optimize the translations by FFT for each one of those rotations, or we can search over the translations by hand and optimize over each one of those translations, or for each one of those translations, optimize over the rotations using FFT. You can also do scoring of fixed poses, but like I said, I'm not going to talk about that too much. There's a number of pre-computations involved in this procedure, right? So, the previous slides, there's been a lot of, expressions for the value of the scoring function as a function of the rotation, as a ligand graph, the conformer, so on and so forth. So what are actually the concrete things that we need to compute, and how often do we need to compute them? So again, it depends on the procedure. So the neural network needs to be run once for every unique ligand conformer, and it needs to be run once for every unique protein structure, right? But it does not need to be run for every combination of protein and ligand. And so this is a distinguishing factor from existing scoring functions, which need to be run, there needs to be a forward pass, at least for every protein ligand complex, and maybe for every candidate pose. So the neural network is only ever run once. I think there was a typo. I meant to say run once here. If the procedure is translational FFT, then you need to compute the Fourier coefficients for the protein once, and for each ligand rotation, right? Because you're doing manual grid search over the rotations, this means that you do need to compute the translational coefficients as many times as there are rotations. Now you could rotate the protein, but the ligand is smaller, and so in practice we're going to rotate the ligand, and for each orientation of the ligand we're going to compute the translational Fourier coefficients. The rotation FFT has exactly the converse procedure, right? So you need to pre-compute the global spherical harmonic expansion for each ligand and for each possible translation of that ligand relative to the protein. And so what this really amounts to is that you take the ligand and you do its expansion around its center of mass, and then the protein, you're going to choose all the sites on the protein where you think the center of mass could be, and you're going to compute the spherical harmonic coefficients, the global expansion coefficients of the protein scalar field within some radius around each possible translation site. So, as this verbal description might suggest, there's amortization possible at several levels of abstraction. So for example, if you have a library of ligand opposes, or sorry, a library of ligand conformers, and then a single protein, then one of these procedures will be more beneficial than the other because some of these procedures are happening at the ligand level and some of the procedures are happening at the protein level. So this here is just a table. It's directly copy pasted from the paper. I'm not going to go through it row by row because I understand there's maybe quite a bit of information here. But just to take a high level example, if you're doing virtual screening, then you have a single protein, right, or a single protein pocket, and you have many, many ligands. And so what you want to do here is the procedure that requires the fewest ligand precomputations, right? And so that's going to be the rotational procedure, because the rotational procedure requires only a single ligand global expansion, whereas it requires many expansions on the protein side. But if you only have a single protein, you can reuse all of them, right? So there are fewer ligand precomputations if you use rotational screening, and so that makes it make more sense for virtual screening. If you're doing inverse screening, you will have the opposite conclusion for essentially the analogous reasoning, except with ligand and protein flipped. Conformer docking is the task that we're focusing on in the talk so far. As I said, it's not necessarily the most standard way that you would do docking because you would likely want to be able to search over the conformational degrees of freedom. Unfortunately, it's just a limitation of the method right now that you can't really do that. So we're going to evaluate against baselines that are also given the ground truth conformer, right? So in all of these experiments, our method and then the baselines are given the ground truth conformer, and we want to see the success rate as R must D below 2. So we're going to have two data sets, one of which is PDB bind, which is of course very well known. The downside of PDB bind is that because all of the complexes have unique proteins and unique ligands, there's actually no opportunity for amortization. And what we're going to see is that the method doesn't really have much of an advantage over standard methods on PDB bind because these precomputations, if you have to do them every single time, they actually do turn out to be quite expensive. But then we're going to look at another set called PDE10A. This is a protein called phosphodiesterase 10A, where there are 70 available crystal structures where these 70 different ligands are docked to the same pocket. And so what this allows us to do is to explore the impact of protein level amortization. And we're going to see that our method is going to have some runtime improvements. So this is essentially the main result table. It's abbreviated quite a bit, but the further details are in the paper. So what we can see is on PDB bind, on crystal structures, we drop a little bit of performance relative to standard scoring functions Vina and Nina. And this is in some sense not unexpected because we are choosing a much more restricted parameterization of the scoring function relative to the pairwise scoring functions in Vina and Nina. On ESM fold structures, docking to these apo structures, we do a little bit better. Again, this is typically expected with machine learning based methods because they're a little bit more robust to sidechain errors. But the key column here is the runtime, right? So the runtime for our method, we report, the first number is if you only look at the pre-computations if you only look at the core computations that cannot be amortized, right? So these are the actual Fourier transforms themselves. How long does that take? And then the second number is how long does the entire procedure take if you include the pre-computations? And what we see is that if you include the pre-computations, which in the PDB bind case cannot be amortized, you actually do not really get a speed up. And that's just simply because there's not very much to amortize as a result of the, each protein ligand complex has a unique protein and a unique ligand. The take home message here is that on PDE10A, actually, let's see, what's going on here? Oh, yes, okay. So the numbers are the total runtime on the right-hand side, and then the core runtime on the left-hand side, right? So in the PDB bind case, the core runtime is quite high because there's no amortization. And then when we go to PDE10A, you observe that the core runtime, the actual fast Fourier transforms themselves, they still take about the same amount of time as expected. And then for the translational case, the translational procedure, the total runtime, it still remains roughly the same because as we discussed earlier, in the translational case, you need to do new pre-computations for every ligand, right? So in the PDE10A case, since you have a library of different ligands, that still ends up being expensive. But the rotational optimization case, because you do pre-computations at the protein level and you can reuse all of those pre-computations, the 68-second total runtime per protein ligand complex, it plummets to 1.5 seconds on PDE-10A. And so this illustrates the benefits of amortization. And as you have more and more and more and more ligands, this 1.5 second per protein ligand complex will asymptotically approach 0.5 seconds, right? Because that is actually the core runtime for doing the FFT. And if you were doing inverse screening, you would see the opposite trend, where the translational procedure decreases in runtime, but the rotational procedure stays constant. So going to the conclusions, what we have here is a method which we have applied to a relatively restricted docking task, right? Just the task of rigid conformer docking. So I would characterize this as a first attempt for using machine learned scoring functions that allow for FFTs. But for this first attempt, we see that the results work reasonably well. For the task that we consider, we do get a favorable runtime trade-off. Again, the main limitation is we're not able to account for confirmation degrees of freedom. So right now, we would still have to use brute force methods. So maybe what you could do is you could instead integrate some conformer generation methods from the literature, or you could do classical optimization algorithms over the confirmation degrees of freedom, but then kind of use this FFT procedure to marginalize over the other degrees of freedom given a particular confirmation. And maybe there are some clever neural network tricks that you can do to avoid rerunning the neural network for each conformer, which would again cut down the cost of searching through the conformational space. Another thing that you could do to further improve the procedure is to have something a little bit more intelligent than doing grid search over the degree of freedom that you're not doing the FFT over, right? Because right now what we're doing is, if you're translating with FFT, you're grid searching over rotations. If you're using FFT to search over rotations, you're doing grid search over translations. And so, maybe you could, alternate, do one after the other, or you could do some sort of multi-resolution setup where you, first do the FFT on a coarse grid, and then you do FFT on a finer grid once you kind of, hone in a bit on the exact placement of the other degree of freedom. So again, these are various variations that you could try out with the method. But what you want to do is just to explore the possibility of learning scalar field-based scoring functions, which is something that at least doesn't seem to have been explored so far in the literature. So so in conclusion, thanks very much for your attention, and happy to take any questions.